Welcome to Vital Biology. This is Padma. In a previous class, we have discussed about two types of the blood cells. One is the RBC, another one is thrombocytes. And in this class, we will discuss about the third variety of a blood cell, that is of the WBC. WBC means white blood cells, which are also called as leukocytes. Okay, WBC is also referred as leukocytes. So, why these cells are called as white blood cells? Because as they lack the hemoglobin in their structure. Okay, if we observe in the case of the RBC, there we have studied that RBC contains hemoglobin, which give red color, whereas such type of a hemoglobin or any other type of pigments are actually absent in case of these cells. So that is why these cells are colorless, and that is why generally they are referred as white blood cells or otherwise leukocytes. And coming towards the structure wise, as the other cells, other eukaryotic cells, yes, the nucleus is present in these particular cells. So that's why these cells are nucleated. Okay, nucleus is present. And in case of their number point of view, if we will see, out of all the three type of the blood cells, that is RBC, thrombocytes and these WBC. WBC are fewer, that means less in number, as RBC are in millions, whereas platelets they are in lakhs, whereas these are in thousands. Okay, in 1 cubic mm of blood, the number of WBC is about 6,000 to 8,000. Yes, that is 6,000 to 8,000 cells per 1 cubic mm of blood. That is why out of three type of the blood cells, these are less in number. But whenever there is any infection in our body, then during those conditions, yes, definitely the WBC number will increase than whatever the normal range which we have discussed here. So such a condition is called as the leukocytosis. Leukocytosis. Leukocytosis means whenever there is an increase in the number of WBC than this normal range. Okay, if it is more than this average number, then definitely the type of a condition is called as the leukocytosis. And when the number of WBC is less than the normal range, okay, if it is below than this normal range, then the condition is called as a leukopenia. Okay, leukopenia. So these are the two terms which we have to remember regarding the number of WBC. Increase than the normal range, that is called as the leukocytosis. When it is less in number, then it is called as the leukopenia. So along with these two terms, even we use one more term which is related to this particular WBC. So that is leukemia. Okay, leukemia. Leukemia is also referred to abnormal increase in the WBC. Okay, abnormal increase in the WBC, and this WBC is immature WBC. Okay, and this is what the normal terms will refer it as a blood cancer. Blood cancer is also called as the leukemia, where abnormally immature WBC is seen, and that is called as the this particular, that is leukemia or otherwise blood cancer. Okay, so these are the different terms related to the number of WBC. So going towards the other characteristic feature of the WBC, that is a lifespan. So what is the lifespan of the WBC? So the lifespan of the WBC is very short. Okay, as they have a different varieties of WBC, they'll range from few hours, few days and a few weeks. And in case of some of the cells which are considered as the memory cells, which keep the memory of the pathogen for several years also sometimes. Okay, but if we will keep the memory cells aside, then if we in general terms, if we want to discuss about the lifespan of all these cells, yes, like few hours, days and weeks also we can see. And going towards the types of the WBC, yes, these WBC can be differentiated into two main groups on the basis of especially the presence and absence of granules. Okay, granules. So generally, if we will take any of the cell, okay, any of the cell if we will take, Generally, cell contains a cell membrane, and internally it is provided with one of a nucleus and a fluid part which is present, so that is the like cytoplasm. Okay, so like this. Generally, this is the normal cell structure. But suppose in this cytoplasm, if these WBC contain a specific granules, okay, okay, then such type of the cells are called as granulocytes, okay, granulocytes, okay. If the cytoplasm contains granules, so granules means, just imagine, suppose if we will take a glass of water and if we will pour a certain like a few of the mustard seeds in it, okay, so like that the granules in this particular like WBC, they are nothing but the some vesicular structures, and these vesicles contains some of the enzymes and some of the antimicrobial agents which helps in killing of the pathogens. So such type of the materials are packed in these vesicular structures, and those vesicular structures are nothing but the granules, and such a granules can be seen in the case of this particular variety of WBC. That is why these cells are called as the granulocytes. Suppose if such type of the granules are actually absent, okay, the cytoplasm doesn't contain any granules, it's just a clear cytoplasm, then such type of the WBC are called as the agranulocytes. Okay, so like these cells can be, WBC can be differentiated into two main categories, that is of granulocytes if the granules are present. If the granules are absent, then such type of the cells are called as the agranulocytes. Okay, and again based on the number and the size of the granules, okay, inside the cytoplasm, these granulocytes are further differentiated into three more varieties. So one is neutrophils, basophils, eosinophils. Okay, neutrophils, basophils, eosinophils. These eosinophils are also called as acidophils. Eosinophils have one more name like acidophils. Like this, three different varieties can be seen in these granulocytes. So generally, if you observe carefully, in each of these cell name, we can like identify one particular character. That is, it is somewhat explaining the pH nature, okay, the nature of the like cell. Here it is describing. Like a neutrophil means generally, yes, it is referring as a neutral. Basophil, basic, whereas the eosinophil are acidophil, acidic. Okay, so what are these terms are referring to? So these terms are referring to those like cytoplasmic contents or the cytoplasmic granules, and these cytoplasmic granules like can absorb some of the chemicals or stains, and if they absorb the stains which have a neutral like a pH 7, okay, pH 7, then such type of the cells are called as the neutrophils, as they absorbing neutral dyes, neutral stains. Whereas basophils means, yes, they absorb basic dyes whose pH is above 7. Such type of the stains, suppose if they will absorb, yes, such type of the cells are called as the basophils. And suppose if they absorb acidic dyes, then so those cells are called as the acidophils. So like this, based on the type of the stain or the dye which they absorb, based on their chemical nature, these cells are got these names as neutrophils, basophils and eosinophils. Generally, basophils absorb dyes like basic nature, and such type of the basic dyes are like methylene blue. Okay, such type of the basic dyes are methylene blue. So this is one of a basic dye, example for basic dye. Whereas the eosinophils are the acidophils. Yes, here you can see this is one of the acidic dye, that is of the eosin. Eosin is the acidic dye. As this eosin is the acidic dye and these cells are absorbing such type of the eosin, that is why these cells got the name as eosinophils. Okay, eosinophils, as eosin is a acidic dye which they are absorbing. Okay, like this. Then what is the example for neutral dyes? So for neutral dyes, the example is like Giemsa and Leishman. Okay, Leishman stain. These are the neutral stains. So that's why, suppose in a lab, if we want to observe these different type of the WBC, like in a one experiment itself, if we want to observe all the variety of the cells, then better we will go with this Giemsa and as well as the Leishman stain. Then yes, it contains both these. Okay, actually this Leishman stain, this contains both methylene blue and as well as the eosin. That means it contains both the acidic and the chemical nature content, and as it is maintaining the neutral value, the neutral pH, that is why such type of the stains generally we use for the observation of the WBC. Okay, so that is about these cells. And these all cells contains the granules in their cytoplasm, whereas here granules are actually absent. Okay, that is the main difference between these granulocytes and agranulocytes. And going towards the number wise, okay, individual cell number, if we want to study, then out of all these cells, okay, out of all these cells, so here neutrophils are like approximately, they are about 60 to 65%. In overall WBC, whatever we said, 6,000 to 8,000 cells per cubic mm of blood, in that these neutrophils constitute about 60 to 65%. Whereas the basophils, these are of 0.5 to 1%. Okay, 0.5 to 1%. Whereas the eosinophils, so eosinophils are about like 2 to 4%. Okay, eosinophils are about 2 to 4%. Whereas going towards the agranulocytes, in which we said two of the varieties are present, lymphocytes and monocytes. Okay, so lymphocytes are again, they constitute about 20 to 25%. Whereas the monocytes constitute about like 6 to 8%. Okay, so this is the average number or the percentages of the various WBC, and all together they'll make around like 6,000 to 8,000 cells per cubic mm of blood. Okay, and going towards their cell structure, shape and function. See here, these three variety of the cells. So these are the granulocytes. Okay, so these are granulocytes. Okay, so granulocytes. So what are the granulocytes which we have discussed earlier? So one is like neutrophils. So this is a neutrophil, and this is the eosinophil, and this is the basophil. Okay, so all these three are the granulocytes. As why they got the name as the granulocytes means, as we know that they contain, okay, they contain granules, that is vesicular structures in their cytoplasm. Okay, so that's why they are called as the granulocytes. Even the eosinophils also contain these granules. But generally, the size and the number of granules are again vary in all these three varieties of the cells. But yes, definitely all the three contain the granules. But going towards the another variety of the cell, which we said agranulocytes. Okay, agranulocytes. So in these agranulocytes, the one of the variety is like monocyte and lymphocyte. Okay, monocyte and lymphocyte. And here you can see that in these two varieties of the cells, generally it doesn't contain, these cells doesn't contain any sort of the granules. Okay, the cytoplasm is clear. Clear cytoplasm is present. Okay, they just contain the nucleus, that's it. They don't possess any sort of the granules. And going towards again these granulocytes. So first one we said neutrophil. So why they got the name as a neutrophil? As they absorb which type of the dyes? Yes, they absorb the neutral dyes. Yes, that is why they said as neutrophils. And these cells have generally, here you can see the nucleus is not one uniform structure. Okay, it's not a one structure. It got segmented. So here you can see this is a one segment, this is one, this is one, this is one. Like this nucleus got segmented. Such type of the segmented nucleus generally will refer it as lobed nucleus. Okay, each of the segment we are considering here it is as a lobe, one lobe. Like that here you can see many lobes. Generally 6 to 8, sometimes even 6 to 10, even sometimes up to 12 lobes also we can see in the neutrophils. Okay, so nucleus is, that is why we'll say multi-lobed. Okay, nucleus is multi-lobed. So here like this, nucleus is multi-lobed. So next, all these multi-lobed structure, multi-lobed nucleus, okay, as even though they are provided with the various segments, but still you can see a small connection is actually present, which is like connecting all these lobes. Such type of the connection is called as the isthmus. Okay, isthmus. Isthmus is the connection. Isthmus is the connection between these lobes. Okay, and these neutrophils are actually the range which we have studied, that is about 60 to 65% in the overall blood. And these neutrophils are also called as polymorphonuclear cells. Okay, these cells are also called as polymorphonuclear cells. Okay, once again I'll draw this one. Yes, so here these neutrophils are also referred as polymorphonuclear cells. Why they got the name as the polymorphonuclear cells? As here you can see the nucleus is divided into different segments, okay, multiple lobes. That is why here poly means many, okay. Nucleus is segmented into various number, okay, various number of segments are present in the nucleus. So that's why polymorphonuclear cells. Generally this polymorphonuclear cells, that is a PMN, this is the short form generally while we'll write to describe this granular, that is of the neutrophils. Okay, PMN cells, that means polymorphonuclear cells. Generally this polymorphonuclear cells is used for the neutrophils, but sometimes this particular PMN is used even for neutrophils, eosinophils and even for the basophils. Okay, as here also you can see the nucleus is segmented. Here not that much partition we can see, but still the term generally sometimes used for all the three type of the cells, that means for all the granulocytes. But here, yes, definitely for the neutrophils. Okay, neutrophils, those are all polymorphonuclear cells. And going towards their function, generally these neutrophils exhibit one of a process which is called as phagocytosis. Okay, phagocytosis. Phagocytosis means generally whenever these cells are, okay, like how the police actually go for patrolling, okay, to find out any sort of crimes or the thefts and all, like that these neutrophils also always they circulate in our body and monitor for any sort of the germs or the foreign substances in our body. So whenever they'll find out any of the pathogen in our body, immediately what they will do is, suppose if this is the, yes, so this is our this is a neutrophil. Okay, so what it will do is, this neutrophil will, so this neutrophil will form one of invagination. Okay, just it will form a one mouth like structure. It opens its mouth and then it is like taking off that pathogen inside. Okay, and slowly it closes that part. It closes that or encloses that particular pathogen. Okay, it enclosing that entire pathogen, and there this pathogen will get digested into small pieces. Okay, so such a process is called as the phagocytosis. Okay, in a simple terms, phagocytosis is nothing but like eating the pathogen by the cell, and there it is digesting inside its cells. Okay, so such a process is called as the phagocytosis, and such type of the phagocytosis is exhibited by these particular neutrophil cells. Okay, and these are the first cells which reach to the site of infection. Okay, so whenever there is any sort of like attack by the germs, okay, then the first variety of the cells which reach to that site of infection are nothing but these neutrophils. Okay, so this is about the neutrophils. Okay, neutral dyes they'll absorb. The nucleus is multi-lobed, and the lobes are connected by one of a connective tissue strand like isthmus, and number wise 60 to 65%, and generally they are referred as polymorphonuclear cells as their nucleus contains many lobes or segments. And how they WBC, like yes, how the WBC generally the function. So whether it is of a granulocytes or the agranulocytes, generally what these WBC will do. So WBC main function is fight against with germs. So but one variety of the cell to the another variety, the way how they fight with the germ is different. So here in the case of the neutrophil, they fight with the germs in a one of the mechanism, through one of a mechanism which is called as a phagocytosis. Okay, phagocytosis. That means it is taking that pathogen directly into its structure, and there it is getting digested. Okay, so phagocytosis, and these are the first cells which reach to the site of the infection. So those are about the neutrophils. And going towards the second variety of the cell, which are eosinophils. Okay, the second variety of the cell are the eosinophils. See, eosinophils. So what these eosinophils? So you're going towards these eosinophils. So here our eosinophils. Okay. So what about them? So these eosinophils are generally contains. So yes, first we will study about the dye, as here also we wrote as they are actually absorbing the neutral dyes. So here eosinophils absorb which type of dye? Yes, definitely it is of the acidic dyes they will absorb. Okay, and generally in the labs the stain which they actually use, so that is of the eosin. So that's why acidic dyes are otherwise the eosin. And here we said multi-lobed in the case of the neutrophil. So here how many lobes here you can see? Yes, 2 lobes. Generally 2 lobes. Okay, and here also we can see one of a connection here. So that connection is the isthmus. Okay, isthmus. And the number wise which we have set about the neutrophils, so that is of 2 to 4% in overall WBC. And going towards the cells, these cells are actually contain granules, and these granules contain a certain substances, enzymes, and these substances are the enzymes are especially contain like some of the toxic materials, and these toxins are act against like some of the parasites. Okay, suppose if any of the parasite means, yes, we know like ascaris, okay, Taenia solium and ankylostoma. So if such type of any of the parasites, or otherwise like Malarian parasite, Plasmodium, okay, so any such type of the parasite, suppose if it is like absorbed by these eosinophils. So these eosinophils have some of the like chemicals in their pockets. The pockets are nothing but here the granules, and from those granules it releases all those toxins and they act against these parasites. So that's why eosinophils play a crucial role in parasitic infection. That is their main function. Okay, eosinophils mainly participate in the parasitic infections. Okay, and even these eosinophils play a major role during these allergies. Okay, In allergic reactions also, mainly these eosinophils play a crucial role. Okay, so that is about the eosinophils. Going towards the third variety of the cell, so that is of the basophil. Okay, so basophil. So the stain which they absorb is the basic stain, and nucleus here if we will see, so the nucleus is somewhat S shape. Okay, may not be a perfect S shape, but generally we will refer the shape of the nucleus as S-shaped nucleus. And the number of the basophils is very less. These are the least number of the WBC, we can say about approximately 0.5 to 1%. And these basophils, so these basophils generally they release some sort of chemicals. Yes, here whatever the granules we said, these granules in the case of the basophils, they packed with some of the chemicals. Okay, so the chemicals like histamines, heparin and serotonin. Okay, histamines, heparin, serotonin. So such type of the chemicals are packed inside these granules. So what is the role of these granules or these chemicals? So before going to that, yes, first we should know the main function of these basophils. So basophils main function is they mainly involves in inflammation reactions. Okay, they mainly involves in inflammation. Okay, yes. So in a simple terms, if we want to understand about inflammation, so inflammation can be like characterized by few of the terms we can use to describe the inflammation. One is like redness, next like swelling, next like itching or pain, and then a slight temperature range. Okay, so generally any of the body part, suppose if we have to say that part is inflamed, so on what basis, on what characteristic features we can say or use the word inflammation means if that particular region is exhibiting the color change, that is into the red color, and compared with the normal other body parts, suppose if it is like swollen, so the swelling character, the next even it results in itching or sometimes the pain, and when compared with the other body parts, the part of the inflammation, yes, definitely it is a little more temperature it exhibits. So all these characteristic features are generally are the main points for the inflammation. And for this entire process, okay, these are the chemicals which are playing a major role. Okay, redness, swelling, itching, temperature. So all these we can say these characteristic features are maintained by these chemicals which are released by the basophils. How they are involved in this particular characteristic features we will see. So the first one we said histamines. So what are these histamines? So histamines are actually called as vasodilators. Okay, histamines are vasodilators. Heparin. So heparin is actually anticoagulant. Okay, it is one of a anticoagulant. And serotonin. Serotonin is a vasoconstrictor. Okay, I'll explain how these chemicals involves in this inflammation. Okay, so now suppose here we'll take one of the region. Suppose here this is the, yes, suppose here this is the one of a place where the damage occurs. Okay, that is the damaged region. And suppose the blood vessel like this which is carrying blood to that particular site of injury, suppose. Okay, site of injury, infection, so in that region, and that site where the like pathogen is present or a damaged or injured region is there, yes, damaged cells, they'll give some sort of signals. By receiving those signals, okay, by receiving those signals, as we told that which of the cells will first reach to the site of injury or the infection, yes, we already have discussed. So those are neutrophils. Okay, so when these neutrophils and even some other cells like eosinophils and all, when they got this particular like signal that there is some damage or injury or infection is going on or pathogens are attacking, then immediately these cells, after receiving that signal, they try to reach to that particular place. Okay, so when they are trying to reach, okay, the more number of cells they want to like reach to that place. So to increase that movement of these cells, okay, so obviously whatever the like pathway or the way which is present, that is may not be sufficient. So that's why the blood vessel, it has to now increase in its size. The diameter of the blood vessel should increase. Okay, like for example, like this, the blood vessel, it is increasing its size like this, so that more amount of the blood, along with that blood, or more amount of the blood cells can reach to that site of injury. Okay, so increasing of the diameter of the blood vessel is called as actually vasodilation. Okay, vasodilation. Okay, and the chemicals which are helpful in that vasodilation are called as the vasodilators. See here, histamines are the chemicals which are released from the basophils, helps in increasing the size of the blood vessel, act as the vasodilators. Okay, and then here, like whenever that particular part is like a little bit, their internal maybe some damage may occur, yes, at that time, to avoid the coagulation process, a free flow of blood should be reached to that particular site of injury. So that's why without any coagulation, the blood in a fluid form, it should reach to the cell to like help in that like a free flow of blood. Definitely one of the anticoagulant, that is of the heparin, is playing a major role. So heparin prevents the clotting of blood. It maintains the fluid nature of the blood, and that fluid nature helps in free flow of all these cells to the site of injury. And the next, so yes, obviously when all these more amount of the blood reaches to that particular region and the cells are more it is present, we can see all these characteristic features, redness, swelling. Because of these chemicals, yes, we'll get that irritation, that is itching or otherwise pain, and even a slight temperature range also we can see. When there is a more number of cell, metabolic activity is also more in that particular place, that's why slight temperature also we can increase in that, we can see in that particular place. And once that like work got finished, now again that blood vessel, it has to come to the normal size. That is why a vasoconstrictor, okay, where it is decreasing the narrowing of the blood vessel should occur, and for that serotonins are helpful, which are called as the vasoconstrictors. Okay, so like this, such type of the chemicals are packed inside the granules of basophils, and basophils playing a major role in the inflammation responses. Okay, the same sort of the function is carried out by one more variety of the cells in the tissues, and those are of the mast cells. Okay, if it is in blood, basophils plays a crucial role in the inflammation. Whereas if it is in tissues, yes, mast cells are playing a major role in that particular inflammation. Okay, so these are about the various type of the, that is blood cells, that is of the granulocytes. Okay, these are all of the granulocytes. Going towards the agranulocytes. Okay, the another variety of the cells which we have discussed, those are of the agranulocytes. Agranulocytes. So agranulocytes means, yes, definitely granules are absent in the cytoplasm. Granules are absent in the cytoplasm. Okay, granules are absent in the cytoplasm, and two variety varieties of the cells we have discussed here. One is the monocyte. So one variety is the monocyte, and another variety is the lymphocytes. Okay, monocytes, lymphocytes, monocytes and lymphocytes. So what are these monocytes? So generally monocytes are like they are the larger one. Okay, out of all the WBC, these are large. Okay, these are large, and these cells have a nucleus, and that nucleus is somewhat a bean shape or a kidney shape. Okay, bean shape or otherwise kidney shaped nucleus is observed. Kidney shaped nucleus is observed. Okay, kidney shaped nucleus is seen, and number wise we said these are about like 6 to 8% in overall WBC, 6 to 8%. And these monocytes also exhibit one of our main important process called as phagocytosis. As we have said, neutrophils, yes, neutrophils and these monocytes have the similar sort of function, that is phagocytosis. Simply they engulf the pathogen and they are digesting that pathogen with the help of the various enzymes. Okay, so here these monocytes are also exhibiting such a phagocytosis process to kill the pathogens. And these monocytes, when they reach to the tissues, then they are generally considered as macrophages. Okay, macrophages. And going towards the lymphocytes. Okay, lymphocytes. So generally lymphocytes are round, and the nucleus is generally occupies the majority of its cell. Okay, so the maximum nucleus is occupying the majority of the structure of the cell. So here you can see a very little amount of the cytoplasm only we can observe inside it. Okay, the maximum like area inside the cell is occupied by the nucleus. And these lymphocytes, characteristic features if we will see, yes, we said in them two different types can be seen. Few of them are small and few of them can be large in size also. Okay, small and even the large one. And nucleus is generally round to oval nucleus. Round to oval nucleus is observed. Okay, nucleus shape, especially in the WBC we can see various different shapes of the nucleus. So in case of lymphocytes, yes, round to oval nucleus we can observe. Number wise if we will see, yes, it is about like 20 to 25% of the WBC, okay, are of lymphocytes. Okay, 20 to 25% are of lymphocytes. And going towards their function. Before going to the function, actually these lymphocytes are of actually two types. B lymphocytes and T lymphocytes. Okay, B lymphocytes and T lymphocytes. So especially here if you will notice, yes, before lymphocytes just we have added B and the T alphabets. So what are these alphabets represents? Actually B means here bone marrow. Okay, bone marrow. Whereas the T means here thymus. Okay, thymus gland. And what are these and why we have added these B and as well as the T means, as we have discussed in the earlier class, that all the blood cells are actually like their origin occurs inside the red bone marrow. But here these lymphocytes, suppose after their origin, if still they present in the bone marrow and if they maturation process, their development also occurs in the bone marrow, yes, such type of the lymphocytes are called as the B lymphocytes. Okay, maturation and development, if it occurs in the bone marrow, then B lymphocytes. If maturation and the development occurs in the thymus gland, then such type of the lymphocytes are called as the T lymphocytes. But we should remember whether it is of the B or the T lymphocytes, their origin always occurs in the bone marrow itself. Okay. And if it is of B lymphocytes, yes, they actually release some of the proteins which are called as immunoglobulins or antibodies. Okay, immunoglobulins are antibodies, and these immunoglobulins antibodies, they fight against with the germs. And what are these antibodies? What is their structure? That we will study in the next further coming classes, that is of health and diseases. Okay, whereas the T lymphocytes, so it plays a crucial role in cell mediated immunity. Okay, cell mediated immunity, లేదా immune or otherwise immune response. Okay, immune response. See here, B lymphocytes playing a major role in antibody mediated immune response. That means whenever any sort of the germ or the pathogen enters, the cells are not directly involved. They are releasing certain proteins which are called as the antibodies, and these antibodies go and fight with the germs. That's why it is called as antibody mediated immune response. Whereas the T cells, they directly will go and fight with the germs. So, okay, cells are directly, they are involved in the killing of the pathogen. That's why they are called as the cell mediated immune response. Okay, so like this, both the T cells and as well as the B cells. So altogether, this is how the WBC playing a major role to fight against with the germs. Okay, so we should remember their nucleus shapes, number, and as well as their functions. Neutrophils multilobed, eosinophils are bilobed nucleus, whereas the basophils are S-shaped nucleus. Agranulocytes, that is of the monocytes, bean-shaped nucleus, whereas the lymphocytes oval to round nucleus. Whereas the number wise, if we will see, neutrophils are about like 60 to 65%, whereas the eosinophils are about like 2 to 4%, basophils are about 0.5 to 1%. Whereas agranulocytes, monocytes are about 6 to 8%, whereas the lymphocytes are 20 to 25%. Functions wise, if we will see, neutrophils main function is phagocytosis, eosinophils to fight against with the parasites and especially in the allergic reactions. Whereas the basophils, inflammation reactions. Whereas in the agranulocytes, monocytes are mainly involved in phagocytosis, whereas the lymphocytes are mainly involved in, if it is of B cells, yes, humoral or the antibody mediated immunity, whereas the T cells, those are involved in the cell mediated immune response.